All right, today we're going to talk about Lab 3. So Lab 3 is Introduction to the Light Microscope. So microscopes are great tools that you can use to see all the things that you can't see with your naked eye. So all the little microbes, all the living organisms that are so tiny that you can't see them with your without the help of a microscope. So myself, I am a microbiologist, so that means that's what I do. I do a lot of study of things that are in the microbial world, so that we have to use a microscope to see them. There are so many microbes, it is amazing. Again, you can think of every plant, every animal, every macroorganism, the ones that you can see with your eye. Think of all of those, and then realize there are so many more that you can't even see unless you have a microscope. So for me, that was fascinating. The first time I actually looked through a microscope was when I was in college. I never got to see through a microscope until I made it to college. And when I did, I thought it was fascinating that, again, I'd thought of all of these plants and animals and critters and everything out there that I could see in the world around me, but then there were all these that I couldn't see because I needed a microscope to be able to see those. So what is a microscope? Again, it's a tool that we use to see those organisms. So today what we're going to cover, we're going to talk about how to use the microscope, parts of a microscope, how to set up a slide so you can put a sample specimen on a slide and then get it on the microscope and be able to see this microbial world using the microscope. So to start off with, you should come in, you'll have the microscope sitting somewhere sort of in front of you on your bench. Do be careful, these are expensive pieces of equipment. If you do have to move them, you always carry them with the arm and the base. So you'll have one hand on the arm, one hand under the base. That goes for any microscope you carry. Again, this is the arm up here, this is the base. You always carry it with two hands. Again, we should have them out ready for you to use. They may be still wrapped up with the cord around them. We may have them unwrapped, I'm not sure, it depends on the instructor. I'll probably already have them unwrapped and plugged in, but you'll want to unwrap the cord carefully if necessary and plug it in. The outlets are right here on the end. If it does not work, if your outlet doesn't work, you might have to hit this reset button. If the green light's on, you're good. Sometimes they get popped. To turn the microscope on, you have a switch on the back. So I'm gonna go ahead and just turn it. You'll flip the switch on the back, you'll notice the light should come on. If your light's not coming on, something's with the microscope or with the outlet. Again, check the outlet first, check the switch on the back. Sometimes also the light gets turned down. So over here we have what's called a rheostat. Sometimes it gets turned down too low and you need to just turn that up. So I'm going to start off talking about the parts of the microscope. So I've already mentioned a few of the parts. You also have the parts of the microscope right there in your handout. You'll notice that microscope looks a little bit different than that microscope, but overall the parts are pretty well the same. I already mentioned the arm. So this is going to be the arm up here. I already mentioned the base. This is the base down here. I've also mentioned the rheostat. So that's what can turn your light up and down. I always recommend having the light turned up all the way as much as you can stand. That's going to make it easier for you to see things. But let's do a top down of the microscope. Up here we have what we call our ocular lenses. They have some adjustment. You should not have to adjust those. So just leave those how they are, don't twist on them. What you can adjust is this right here. So if you pull in or out on it, again, depending on how far your eyes are apart, you might need to push in or out. So you can adjust it to where you need to be able to see in there. Before you put your eyes up near them, again, we're going to be trying to be as safe as possible. We're going to have everybody wearing these nice stylish glasses, which will be awesome, especially if you have a hat, glasses, and a mask. That'll be really fun. But if you're wearing a hat, you probably have to turn around backwards like that, but then you should be able to see into the microscope. Where am I? Oh, I'm on this side of it. There we go. So again, look into the microscope like that. But you should have your glasses on first. Again, parts of the microscope. Ocular, that's what you look into. I've told you can adjust it. I've told you need to have glasses on before you do so. So ocular's up here. Then you have the body tube right here. So this is the body tube. That's just where the oculars attached to. You have the arm right here, rheostat right here that turns your light up and down. You have the base down here. You have your stage. This is called the stage. This right here is called your stage clip. So it works like this. You turn it like this, and we'll talk about how we use this to put in the slide. So what the stage clip does and the stage holds your slide, and the stage clip moves over like this, so you can put your slide in. So you don't slide your slide under the stage clip. You open it up by pushing it like this, put it into that nice little pocket over there, and then close it. So stage clip, again, ours opens and closes like this. You don't try to slide it under it. Don't do that. You open it up and then slide it in here. Sits on your stage, so stage clip. These are called your objective lenses. So you'll notice here they spin. The part they spin on, this is called the nose piece. So this right here that I'm spinning, this is called the nose piece. And each one of these is called a objective lens. So these microscopes have 4 objective lenses. They differ in their magnification. So whichever one you have turned down facing the slide is the one that you have selected. So if you notice, that one says 4x. That means it has 4 times magnification. Now this one here has 10 times magnification. And this one here has 40 times magnification. We're not going to use this other one, but it has 100 times magnification. If you'll notice, it also says oil. We have to use a special oil called immersion oil to use that one. We're not doing that in this lab. But we're going to use these other 3. So we're going to use the 4x magnification, the 10x magnification, and the 40x magnification. So again, I said this is 4 times magnification, but if you're looking at a sample with that 4x, these lenses up here also have magnification. So if we look up here, you'll notice it says 10x. So these lenses have 10 times magnification, and if you combine that with the 4 times magnification, what you do, you multiply those together. So you have 4 times magnification and 10 times magnification, you multiply them together and you get 40 times magnification. So 40x magnification. It's going to magnify that object 40 times. Now again, same with if we have the 10x, you multiply that 10x with this 10x, and you have 100 times magnification. Finally, if you have this high power lens here, this high power objective lens, 40 times 10, 400 times magnification. So we're going to be magnifying things up to 400 times in this lab. So other parts, so to go over it again, we have ocular, body tube, arm, stage clip, nose piece, objective lenses. The other parts we're going to talk about are light source. So again, our light source we adjust with the rheostat over here. We have the iris diaphragm. So this is the iris diaphragm right here. If you notice, what that does is it also adjusts the light. So you've got 2 ways of adjusting light. You can use the diaphragm here that'll close off and adjust the light, or you have the rheostat over here that'll adjust the light. Again, my recommendation is always more light is better. You know, do as much light as you can stand. You know, some people get headaches or have problems with the light, but do as bright as you can. That's going to make it easier to find things. So we talked about the iris diaphragm, stage clips, base. The other important parts are the coarse adjustment and fine adjustment. So you have one there, you have one here. Depends on if you want to use your left hand or right hand. They do the exact same thing. Your coarse adjustment knob is on the outside. So you see what happens when you turn that, it's going to move the stage. What that's going to allow you to do is focus the microscope to be able to see your sample. The fine adjustment gives you fine adjustment on that focus. So the way that you want to always start with, for finding a sample, is you want to start with the low power, the lowest power lens that you have, the lowest magnification. So you're going to start in this case with your 4x objective lens. And what you're going to do is, what I always do is I bring this up as far as it'll go. Again, you don't want to smash your slide into the lenses. So sometimes they get off adjustment a little bit. So just be careful, when you're doing it, that you don't smash it into your slide into the lens. You don't want to break your slide, you don't want to break the lens, but you want to bring that stage all the way up. And then what you'll do is you'll start looking through the microscope. I don't have a sample on here, but you look through the microscope and I won't be able to find it. You'll notice there's a needle in there. That's a pointer needle. It will point to part of your specimen. On some of these microscopes, they have a little ruler. This one I'm not gonna be able to get to show. But ultimately what you do is you adjust your stage downwards slowly by rotating the coarse adjustment knob, not the fine adjustment knob. You're gonna use the coarse adjustment knob. You're gonna rotate your stage down slowly while you're looking through the oculars until you see your sample come into focus. It might not be perfect focus. You're gonna get it as close as possible to focused. So you'll be looking through until you see something, and then you'll move to the fine adjustment knob and get it as clear as possible. So that's all there is to it. Start all the way at the top, rotate it down slowly till you see it, and then adjust it with the fine adjustment knob till it's nice and clear. Now, if you want to increase the magnification, you don't touch any of this. Don't touch any of that to adjust the magnification. What you do is you turn this and lock in the next highest magnification. You might not see your sample perfectly, so what you do in this case, you don't touch the coarse adjustment. You'll use just the fine adjustment to then get it into focus. These microscopes are what are called parfocal, meaning that you should not have to adjust the focus much when you go between different magnifications. So again, you should just be able to adjust this a little bit. You don't want to spin this more than 2 times one direction or the other. If you're having to spin it more than 2 times one direction or another, talk to your lab instructor. They'll help you get it focused. You might have to adjust that slightly, but I recommend not doing that. Typically, all you're gonna have to do is adjust this a little bit and it'll get into focus. Once you get that one in focus, if you're trying to go to the highest power, you just turn it and then just adjust that a little bit. If you follow those rules, you should be able to find things quickly, simply, and easily. We shouldn't have too many of that. I can't find anything under the microscope. I can't see anything. So again, to show again, low power, all the way up with the stage, rotate it down slowly until you see something with the coarse adjustment knob. Once you see it, adjust with the fine adjustment knob. You want to go to higher magnification and just spin it. It's got to actually click into place. You'll feel it click, and then fine adjustment knob again. Now, the one thing I didn't point out is this right here. This is what moves your slide. So you'll notice this one on the bottom moves one direction or the other. The top one moves it on the opposite axis. So again, if you're trying to find something, if you've got your letter E on there and you can't find your letter E, use these to adjust it. Don't try to push with the slide. Again, we have these nice ones that you can adjust like that. The way you know that you're focused on the right thing, again, you're looking at where the light is. So if you're trying to find like your letter E, move it to where your letter E is, right over that bright light. Okay, so covered the parts of the microscope, covered how to carry it. Again, they are expensive pieces of equipment, so do be careful with them. If you have questions about any of that, ask your instructor. I did cover how to do magnification calculations. You will use rulers to figure out what your width of your field of view is. When you use the ruler on it, you literally just put the ruler right on here. So you're gonna have some transparent rulers you're gonna be looking at. You'll put the ruler right on here to get your field of view. The other thing you're gonna be responsible for setting up are your slides. So you're gonna be putting samples on your slides. Turn the hat back around. You have to do that so I can see the microscope. You're gonna have a dropper. Again, all of you should be familiar with these droppers by now, these transfer pipettes. You may be using some different samples. Your instructor will have those out available for you. Your handout tells you which ones to use. Your instructor will give you more information on how to get those individual samples. But again, you're going to use that transfer pipette, same as you did in the last lab. Get a beautiful view of my face there. Again, it's going to tell you to do 1 drop. Make sure you just do 1 drop. You're going to do 1 drop of water. You'll put whatever your sample is on, and then you use these little cover slips. One common mistake is that people don't take the paper off the cover slip. Make sure you take the paper off the cover slip. It should be a nice clear cover slip. And then you just put your cover slip right down on the slide like so. And then you put that right on the microscope. Nice and simple like that. So, again, any questions on anything else, make sure you ask your instructors. That's what we're all here for, is to help you through all of this. Read your handout before coming to lab. Especially, pay attention to these. Your instructor is going to help you with these, guide you through some of these calculations on how to do the metric conversions. But if you have any questions on that, you can reach out to any of the instructors. I'm sure any of them would be happy to cover some of that with you. With that, I think that pretty well covers for Lab 3. Again, the main thing is covering the parts of the microscope. Make sure you get those. And if you follow those steps on how to find the image of, start with the low power all the time, start with the low power, have your stage all the way up, slowly rotate that coarse adjustment knob to where the stage goes down while you're looking in there and you can see it, and then use the fine adjustment knob. When you go to switch magnifications, don't touch either of those knobs. Don't try to restart it. If you try to restart it, you'll get out of focus, and that won't be good. Again, get your focus with the lowest power, nice and clear, and then switch to the medium power, focus it, switch to the high power, and focus it. If you follow those rules there, you'll do great with it. The other key, again, is keep the light as bright as you can stand it. That'll definitely help with that. Okay, I think that covers everything. Again, your instructor will tell you where each of the things are depending on the way they set up the lab. It'll be similar as before. We'll have things set on the side benches for you to grab the different samples. The other key is when you're getting the little amoeba and paramecium and so forth, mix up the sample good. So swirl up. Sometimes it's good to stir it. There will be little floating things in there. Sometimes good to get those little floating things and put onto your slide. Again, you just want 1 drop though. With that, I think this video is long enough. I get excited about microscopes. But anyways, everybody have a great week. Good luck with lab.